You are once again welcome to Bio 211D, Plant Physiology, Unit 6, Session 1, Stomatal Structure and Movement. And by the close of this session, students should be able to describe the structure of the stomata, name the parts of the stomata, state factors that control the opening and closing of the stomata, and explain how stomata control laws in plants. Stomata are the primary control organs or structures that plants use to reduce water loss. They are able to do so quickly to prevent the plant being starved of water, which is essential for various activities. Note that water serves as a medium of biochemical reactions, transport, as well as a raw material for photosynthesis, among other things. So stomata are sensitive to the environmental signals that trigger the stomata to open or close. The major role of stomata is to allow carbon dioxide entry, to drive photosynthesis, and at the same time allow the exit of water as it evaporates, thus cooling the leaf. So two specialized cells called gas cells make up each stoma. Plants have many stomata, up to 400 millimeters per square on their leaf surfaces, particularly on the lower surfaces. This arrangement is to minimize water loss. The structure of stomata are part of the leaf epidermal tissue. So each stoma is bordered by two guard cells, which are kidney-shaped. The inner cell walls are thicker than the outer cell walls. Thus, the outer cell walls expand faster than the inner ones when the guard cells are turgid, thus creating the stomatal aperture or pore. Each guard cell, like any other epidermal cell, has a nucleus, a vacuole, and other cellular organelles. So in here we have the structure of the stomata, whereby we have the epidermal cell, the stomatal pore, thin outer wall, and we have nucleus, and these are the guard cells. Then the chloroplast itself, we have sub-vacuole, thick inner wall, subsidiary cell. So these definitely shows the structure of a stoma, showing guard cells in the stoma or stomatal pore. So as stated earlier, environmental cues influence the opening and closing of stomata. Environmental factors that affect opening and closing include the sunlight, water temperature, concentration of carbon dioxide within the leaf. Light. Stomata usually open in the light and close in the dark. Why should this happen? This is because plants use solar energy to produce organic compounds, that's glucose, from simple organic compounds such as carbon dioxide and water. Hence, the stomata must open in the daytime to allow the intake of carbon dioxide. However, stomata can close in the middle of the day if water is limited or carbon dioxide accumulates in the leaf or temperature is too high. If a plant lacks adequate water, stomata will close because there will not be enough water to create pressure in the guard cells, which control their opening for stomatal opening. This response is necessary for the plant to conserve water. So internal concentration of carbon dioxide. When the internal concentration of carbon dioxide of a leaf increases, the stomata are signaled to close. This is because respiration is releasing more carbon dioxide than photosynthesis is using. Therefore, there will be no need to keep the stomata open and lose water if photosynthesis is not functioning. So on the other hand, if the internal carbon dioxide concentration is low, then it means that the stomata will stay open to continue providing carbon dioxide for photosynthesis. Temperature. High temperatures will also signal stomata to close, since such temperatures will increase water loss from the leaf. With less water available, guard cells become flaccid and close the stomata. High temperatures also cause respiration rates to rise above photosynthesis rates. Consequently, internal carbon dioxide concentration in the leaves also increases. So the high internal carbon dioxide will then cause stomata to close as well. However, some plants may open their stomata under high temperatures so that transpiration will cool the leaves. Stomatal movement. The opening and closing of stomata are referred to as stomatal movement. For stomata to open, potassium ions enter the guard cells. This results in increase in osmotic pressure in the guard cells. The osmotic pressure causes water to enter due to the low water potential created by the potassium ions, causing a water potential gradient. The entry of water creates hydrostatic pressure in the guard cells, thus changing the shape of the stoma. Guard cells expand on the outer edges of the stoma, but not on the inner side. This unequal expansion results in kidney-shaped cells, thus opening the stomata or creating a pore between the two guard cells for gas exchange. Kidney-shaped guard cells are characteristics of dicots. Many plants such as grasses and other monocots have dumbbell-shaped stomata. The shape taken by the guard cells is dependent on cellulose microfibrils that spread out radially from the pore, somewhat similar to radial ties. The cellulose microfibrils are rigid and do not stretch when water enters the cell. The cell walls surrounding the stomatal opening are thickened, preventing that side of the guard cell from expanding. Therefore, if pressure in the cells increases due to water entry, the guard cells do not expand. Rather, the outer edge stretches disproportionately more than the inner edge. This unequal stretching allows the pore to form between the two guard cells, thus creating the stomatal pore or opening. Stomata must be open for the plant to obtain carbon dioxide for photosynthesis, as stated earlier. However, open stomata could cause a plant to lose too much water through transpiration. As said earlier, stomata closes when the guard cells lose water and become flaccid. This occurs because the potassium ions move back out of the guard cells, followed by water that lowers the tubal pressure in the guard cells. So in here we have an open and a closed stomata being compared, whereby the guard cells actively take up potassium ions, causing water to enter by osmosis. When the cells of guard cells are unevenly thickened, you have shrunken guard cells. That's when they lose water through transpiration. So stoma that is open is being compared with a stoma that is being closed. So guard cells actively take up potassium ions, causing water to enter by osmosis. The cell walls of guard cells are unevenly thickened, using the cells to become bowl-shaped as they become turgid, causing the stomata to open. When guard cells become flaccid or lose water, the stomata closes. So in review, the physiology of stomatal opening and closing relating to the structure of the stomata were discussed. Then again, factors that control stomatal opening and hence controlling the loss of water from the leaves were also talked about.